Hi learners, good morning to you all. We are now come to the fact end of Unit 1, that is topic 8. The topic 8 deals with antigen processing and presentation. Myself is Professor S. Krupalini, heading the Department of Biotechnology at Vignan's Foundation for Science, Technology and Research, Andhra Pradesh, India. I am giving a series of talks in this. This is the topic 8 of Unit 1. Hitherto, we have seen that in this topic, antigen, adjuvant, heptane and antigen, the factors influencing the antigenicity. So with this, again let us revise what is antigen. Antigen is a foreign protein. Foreign means, what is that foreign? That is phylogenetically ancient protein, phylogenetically distant protein. That is, the species like bacteria, the species like fungi, the species like protozoa, the species like invertebrates, they are all far from mammals, humans. So that's what the protein from them constitute the antigen. So now regularly we keep eating egg proteins, we digest them, we totally fertilize them, and we take the amino acids. Those amino acids are useful in the transamination and transcarboxylation reactions in anabolic metabolism. Okay, so that is altogether different. That doesn't constitute the antigen. But now the foreign proteins, that's whatever I was just now mentioning, phylogenetically distant proteins, if they are perceived and taken by this immune cells called the dendritic cells, then it is being processed. So the details of the process we will be knowing now. Not only the process, it doesn't end with the processing, it leads to the ultimately presentation. That where to present, what to present. This antigen as it is will not be presented. The processed antigen is being presented. So the processed antigen, what is it? It is nothing but a fragment of the antigen called epitope. Okay, the epitope containing approximately 15 amino acids, that is being presented. So presentation, how is it presented? Is there any media? Is there any procedure? So here again, there is MSC, major histocompatibility complex molecules are involved. They carry two T helper cell, T cytotoxic cell. That carrying process is called as the presentation. The details of it we see in this particular topic A. Okay. Now let us see the objective. So to understand the cytosol process of antigen. Cytosol is a cytoplasm of a cell wherein the antigen is being processed. Once the process is completed, then the products of the process are being presented by MSC class 1, class 2. MSC means major histocompatibility complex of class 1, class 2. The class 1, class 2 are distinct proteins. Okay. Just now, we have gone through these objectives wherein MSc class 1, MSc class 2 are involved in the presentation. Now let us go through the expected outcomes of the topic 8 on antigen presentation and processing. So in this, the learners are going to know the development of MSc class 1 coupling with viral antigenic peptide, development of MSc class 2 coupling with bacterial antigenic peptide. Please make a note here. MSc class 1 is being associated with viral peptide. MSc class 2 is associated with the bacterial antigenic peptide. And antigen presentation ultimately elicits immune response. These three aspects are the important aspects of the immune system, and arousal of this immune mechanism will be known. Now let us see.
In the last video lecture we saw how processing and presentation of intracellular pathogens takes place. Today we will study processing and presentation of extracellular or exogenous antigens. As the name indicates, extracellular or exogenous antigens are the antigens that originate from outside a host cell. For example, extracellular bacteria. These antigens are taken inside by cells either by endocytosis or phagocytosis. The cells involved in this pathway are antigen-presenting cells. Recall that antigen-presenting cells are phagocytic cells which present peptides derived from internalized pathogens. These include B cells, macrophages, and dendritic cells. The pathway starts when the extracellular antigens such as extracellular bacteria are internalized by the antigen-presenting cells. Let's zoom in to study this pathway. When an antigen-presenting cell internalizes a pathogen, it internalizes it into an intracellular vesicle known as endosome. In the next step, this endosome fuses with lysosome to form endolysosome. In the endolysosome, the internalized pathogens are digested by proteolytic lysosomal enzymes into small peptide fragments. MHC class II molecules are synthesized and assembled in rough endoplasmic reticulum. Here, the alpha and beta chains of the molecules associate with a third protein chain, called the invariant chain. This invariant chain binds to the groove of MHC class II molecule. This binding stops self-peptides in the rough endoplasmic reticulum from binding to the MHC molecule. These MHC2 invariant chain complexes are transported from the endoplasmic reticulum to Golgi apparatus and from Golgi apparatus to another vesicle. The invariant chain is digested and only a small fragment remain bound to the peptide binding groove of MHC2 molecule. This fragment is known as CLIP. CLIP stands for class II associated invariant chain polypeptide. In the next step, this vesicle containing MHC2 molecule bound to the CLIP fuses with the vesicle containing peptide fragments of internalized antigen. The peptide fragments displace the CLIP and bind to the peptide binding groove. These peptides are 11 to 15 amino acids long. The newly formed MHC2 peptide complex is transported to cell surface. Once on the cell surface, they are presented to T cells. These T cells recognize the peptide displayed by MHC2 molecule by T cell receptor. Another receptor, known as CD4 coreceptor, binds to the beta-2 domain of the MHC2 molecule. Since this pathway involves MHC class II molecules, it is also known as MHC2 antigen presentation pathway.
So just now, we have gone a very nice animated video wherein it is shown a bacteria, later a phagocyte, dendritic cell, and then the dendritic cell processing and presenting through MSC molecule 2. MSC molecule 2 are made in endoplasmic reticulum, that is being protected with invariant chain, and later the peptides of bacteria are anchored, 15 amino acid peptide is anchored to this MSC molecule 2, and then that is being presented to the T helper cell. So that's what the presentation process is shown. And the T helper cell is having the T cell receptor complex. The T cell receptor complex also possesses a co-receptor called the CD8, CD4. The CD4 gets bound to the B2 globulin, beta-2 globulin, that is the beta component of the MSC2 complex, and then presentation is completed. Very nicely it is shown in the video. Let us see the details of this antigen processing and presentation. So for recognition of the antigen, the T cells are required. So in the previous topics we have seen, T cells cannot see the free floating antigens. The T cells can view only those antigens that are presented by MSC class II. exogenous antigen presentation. Now let us see here, this is an antigen presenting cell entrapping bacteria through palms and PRR, and again this bacteria comes to the lysozyme, and this lysozyme is a bag of enzymes that digest the bacteria, and the peptides are being presented to this cells, the T cells, through MSA class 2. So like this is the peptide. So like this the presentation goes, processing. So we have to see in the next few slides the processing, how a peptide comes from an antigen. Okay, now let us see the class 1 MSC pathway of processing endogenous cytosolic protein antigen. Now this is a virus, entrap, it is coming into the phagosome, and again the phagosome is having the bag of enzymes, they digest the virus and the proteins are degraded, and again further the proteins are subjected to a protein mill. That protein mill is called proteasome, and through this proteasome the peptides are regenerated, the peptides are released, and the peptides are passing through TAP, that is transport agent protein, into the endoplasmic reticulum. In the endoplasmic reticulum there is MSC class 1, and into that the peptide gets anchored, and ultimately the peptide gets released out of the cell to the CD8 cell, that is cytotoxic T lymphocyte. So like this, the processing of the antigen coupling with MSC class 1 takes place before it is being presented to cytotoxic T lymphocyte. Okay, next. So now let us see how MSC class 1 molecule is assembled. So a newly born MSC molecule, like that. Now let us see the assembly and stabilization of class 1 MSC molecule. A newly born baby, how mother is protecting. Like that, a newly formed MSC class 1 needs to be protected with molecular chaperones, they are called calnexin. So the calnexin keeps bonding to the MSC, now MSC class 1 molecule. Later, calnexin gets released, and calreticulin and tapacin, these two other chaperone molecules are being attached after the release of the calnexin. Then beta 2 microglobulin is being bound to MSC class 2 molecule. It has become a full MSC class 1 molecule now. Okay, so this MSC class 1 molecule, as we have seen in the previous case, get anchored to the peptide for presentation to T cytotoxic lymphocyte. Okay, now let us see the assembly of class 2 MSC molecule. So class 2 MSC molecule is distinctly separate from that of the class 1 molecule. So the class 2 MSC molecule is the naive molecule that is shown here, that is being protected by a chaperone molecule called invariant chain, and then the invariant chain is being attached to the cleft of the MSC2 molecule, and later the invariant chain is replaced by cleft, that is class 2 associated invariant chain of the polypeptide, and later the clip is also being replaced by the peptide, that is the peptide, antigenic peptide that has been brought to the scene by HLA-DM, and that is being attached and presented. So like this, now let us see in the gross way the dendritic cell. The dendritic cell is having the palm, is being encountering a pathogen having the palms, pathogen associated molecular pattern. Okay, later through PRR, that is pattern recognition receptors, it is bounding to the pathogen. So these two are being processed in the cytoplasm as it is shown in the arrow. Then the processor one, ultimate processor one is the peptide. That peptide is being presented to the T helper cell through MSC class 2 molecule. So now the T helper cell may take the decision whether it is pro-inflammatory activity or anti-inflammatory activity. Okay, so that's what the total presentation means. Now let us see the summary of antigen presentation pathway. This is the antigen presenting cell. Okay, this is the antigen presenting cell presenting the viral peptide through class 1 molecule. As a result, there is endogenous cell destruction. The total of antigen presenting cell is getting destructed because it is carrying the virus part. Now, in the second case, antigen presenting cell takes the bacteria, and the bacteria is taken to the lysozyme, that is being processed and presented to the T helper cell through HLA class II molecule, and then T helper cell presents to the B lymphocyte. The B lymphocyte ultimately produces immunoglobulins. That is the final outcome of what is called antigen presentation. Now let us see the summary. Antigen presentation is a function of MSC molecule. The class I MSC pathway processes endogenous cytosolic protein antigens. Assembly and stabilization of class II MSC molecules are made with invariant chain. Invariant chain is chaperone protein. And let us see the study questions. Explain the class I MSC pathway of processing of endogenous cytosolic protein antigens. Describe assembly and stabilization of class I MSC molecules. Elaborate on the assembly of class II MSC molecules to process antigen. Show a labeled display of antigen presentation pathways. Okay, so with this, we have completed the 8th topic on antigen processing and presentation, and the unit 1 is having total of 8 topics. All of them have been covered. It is quite interesting for us to get introduced to the immune system through this unit 1. Please go through. Thank you, Ananda. Thank you.